Hello and welcome to our lecture on enzymes. So enzymes are an important function of proteins, and oftentimes we refer to this function as one of the most important parts. And one of the reasons why is because of this basic concept, and that is all life functions involve chemical reactions. So chemical reactions are when bonds are broken or formed, and bonds involve interactions between outer energy level electrons, and electrons are energy currency. Energy, which is with the unit G, G just stands for Gibbs, so Gibbs free energy. It's stored within chemical bonds, and when released, it comes out as electrical energy, passing charge from one molecule to another, and as heat. In chemical reactions, the starting materials we usually call reactants, or by biologists, substrates, and the end materials are products. Metabolism is the sum total of all the chemical reactions taking place in a cell. All chemical reactions involve a change in energy. And as I said there, for further info, refer to chapter 2-5. One of the things that we could potentially talk about here with regards to chemical reactions is the fact that the chemical reactions often require too much energy to be added to them to be able to get them to happen in one little step. So they happen in incremental step. So chemical reactions often you're going from substrate A to product B, and then product B is going to be converted to product C, and then product C is converted to product D is converted to product E, and product E is converted to product F. And then product F is the ultimate thing that we are going to want, and then we would call all these intermediates. And all these intermediates are small incremental chemical reactions that don't take as much energy to take place. So you might only change A incrementally, and then you change B incrementally, and then C incrementally, et cetera, until you actually get to the point where the product that you want is formed. So those incremental steps, or taking in a chemical reaction and breaking it into incremental steps, we call those incremental steps pathways. So what are the conditions that are required for carrying out chemical reactions, and how do cell structures meet these requirements? So first off, they need a pH range of usually between 6.8 and 7.4. And the change in pH could affect the molecular structure. We call that change in molecular structure by breaking hydrogen bonds, for example, denaturation. It happens in proteins, and it can happen in nucleic acids. Those are things that are involving hydrogen bonds. They require a specific temperature range. For us, that's 37 degrees Celsius, thereabouts, or environmental temperature. Temperature is a function of molecular movement, and a high enough temperature is needed to ensure interactions. If it's too hot, there can actually be molecular breakdown. If it's too cold, the molecular movement is too slow for interactions to occur, and therefore you don't actually perform the changes in you don't break the bonds that you're supposed to break. So thirdly, they have to maintain substrates in a sufficient concentration. So that's basically what you're doing most of your life, is you are trying to obtain your substrates. You're eating, you're processing. Your food requires digestion, processing, transportation, and for the cells to be ensured a sufficient concentration of substrates. Think about breathing. When you need oxygen, you need to breathe harder when your need for oxygen increases. So these are all examples of ways in which your body and your cells are maintaining these chemical reactions. It's all part of homeostasis. skipping number 2. They just don't want me to go to number 2. There you go. So forming bonds is going to release energy. Breaking bonds is going to consume energy. So although energy is neither created nor destroyed, some of the original energy found in the molecules can be released as heat, and different bonds have different energy levels. So bonds will net in more energy released. There's 2 types of energy released in there's 2 types of reactions. One is an energy releasing, also referred as exergonic, and the other is energy storing, which is endorgonic. And we mentioned this before, but basically an exergonic reaction is where smaller products are made relative to the reactants. Endorgonic reactions are where you have larger molecules formed relative to the reactants. And then you have the concept of activation energy. So you have 2 graphs that represent exergonic and endorgonic reactions. So this is a graph that represents the endorgonic reaction on the left, the one in red. And again, basically what I'm saying is you're forming larger products than you had substrates. So this is going to mean you're going to need to add extra energy in the initial part, but a net amount of heat would be absorbed. So this takes in energy, and then the products will ultimately have a higher potential energy than the reactants did. Notice in both of these cases that a greater amount of energy than what was initially or what was produced is going to need to be added in this reaction. We call that activation energy. So this additional energy is that activation energy. And this is where enzymes come in. What enzymes do is they lower the activation energy. So in the case of an exergonic reaction where a net heat is released, it will release it so that almost no additional energy needs to be added, maybe just the heat of the environment to cause it to go from being at this higher potential energy to a lower potential energy product. And skipping 4. So enzymes work by having specific active sites. Let me actually skip back for a second here because I think So what's important about an enzyme is enzymes are folded proteins that act as catalysts, and they do that by lowering the activation energy. So how do they do that? So they have an active site, and an active site is a cleft in the protein. So what you see here are all different R groups. Okay, so that's an R group, and there's another R group, and those R groups are arranged in a particular way. Sometimes you talk it like a lock and key type reaction where they're able to interact with a specific substrate, and they react with it by forming non-covalent interactions. Sometimes they might have cofactors, inorganic substances that help them carry that reaction out, besides just the R groups. So this is an example of an enzyme called Ribisco, or ribulose bisphosphate carboxylase oxidase. And what it's doing is it's taking carbon dioxide and it's combining with this molecule called RUBP.
This is Dr. Kat. Just wanted to say thank you to all of our
Okay, continuing on. So this is an enzyme, and basically what it's doing is it's forming a bond between carbon dioxide and this molecule called Ribisco. It's a key process in photosynthesis, which we'll learn later in this unit. So what happens is a substrate and an enzyme form a complex. Again, forming non-covalent interactions. And what that does is it produces a transitional form. A transitional form is a form of a substrate where the bonds are stressed so that they can form product. The enzyme itself will then go back to normal after the reaction completes. So typically the enzyme undergoes a change in shape when an enzyme in the enzyme substrate complex, and we call that change in shape an induced fit. And the lock and key theory of how an enzyme works, that assumes a static shape, but once we've been able to actually view once we've been able to view proteins in actual we've been able to view proteins actually in nature. We've been able to see that they have been actually moving around, wobbling. So the active site can be specific for one substrate or many substrates. Some enzymes catalyze the forward or reverse reaction depending on the concentration of the product or the reactant, meaning they can actually form reactant from product or product from reactant, depending on which one there is more of. Okay. So what this is showing is that there is a specific type of an enzyme. So this is an enzyme called sucrase. And this is showing the different steps of the breakdown of sucrose into glucose and fructose. So it says in the lumen of the small intestine, sucrose comes in contact with the sucrase enzyme. Sucrose is going to enter the active site. A non-covalent bond forms between sucrose and the R groups in the active site. This weakens the bonds in sucrose and water, and then sucrose is converted to glucose and fructose. Love the fact that people are back in school. So hopefully the music in the background isn't bothering you too much. Anyway, enzymes are going to usually be there and they're going to be produced. And one of the things that your body does is it produces enzymes, but then it inactivates them. They might make precursors to the enzymes. Enzymes are going to require time, energy, and materials to build. So if a reaction is necessary for homeostasis, but it's not needed all the time, they might form a precursor so that it can really quickly activate the enzymes when they're needed. So how does enzyme regulation done? So really the only way it's done that doesn't have to be done quickly is a long-term developmental process. So bacteria, they do everything this way. They turn on genes and produce enzymes when they need it. So if they don't need the enzyme, they don't produce it. So that's basically how bacteria work. We do that in some regards, but usually you're only doing that during your developmental process. Like when you might be using enzymes only for specific times. One of the times is like when you are actually producing, going through cell division. Like your cell will say, oh, we're going to go through cell division. We're going to produce all the enzymes that we're going to need for cell division now. And then otherwise they're not there. And that's just, it's helpful so that your cell doesn't start doing and producing more and more cells. That's one of the causes of cancer, is basically where your genes that produce that, they're always on. So they're always producing the enzymes that are needed to cause you to go through that division. I'm going to pause and give you an opportunity now to do this so I can also mute the music in the background. Okay, so this last picture is talking about an allosteric enzyme. And allosteric enzymes work as a result of some sort of regulatory molecule. One of the ways in which it works is through what's called feedback inhibition. So if the product is formed here, once enough product is formed, some of the product actually gets converted into the regulatory molecule, which then closes it down. And then once the product is at a low enough level, then the regulatory molecule is taken up by the cell and used as the product. And then some of the product actually then it goes and produces more product to the point where it kind of maintains a balance. So it does this in the case of like a regulatory thing, like a homeostatic function. So then there's synthetic ways. So as it says, organisms have evolved chemical means to either control other organisms' enzymes or even control their own. So humans have learned to harness many of these for pharmaceutical purposes. So a competitive inhibitor is something that will not have a permanent effect because it forms non-covalent interactions, but it basically slows it down. It blocks, it attaches to the active site and can interact with the active site in a way that to prevent the substrate from coming in there. A non-competitive inhibitor is going to be something that's going to form a covalent bond with an enzyme and permanently change it so that it doesn't work anymore. And it can have a permanent effect if it does form that covalent bond with it. Okay. Another way I talked about feedback inhibition is where like an intermediate product could be used to, or I guess the final product can actually block A. So if like A is an enzyme here, and I produce a substrate. So 1 is my initial substrate, 2 is the intermediate. So remember, we're saying that this is step-by-step process. Then 2 is converted into product 3, and then 3 is converted into product 4. And then once I actually have enough product 4, I get enough. Some of the 4 is going to be then used to actually block A to prevent the process from going on. Again, it's kind of maintaining a balance in that way. So these are different ways in which enzymes are inhibited. One of the things you see in this graph is both of these graphs reaching saturation points. So saturation points is where the enzyme stops working any faster, and it stops working faster because you have an active site, so you have a finite area where the amount of enzymes can actually be carried out. So as this says here, normal enzymes reach its maximum rate and then levels off due to reaching saturation with substrate. Adding additional substrate will not change the rate of the reaction. So competitive inhibitors are competing with substrates for active site. So the amount of substrate needed to overcome the inhibitor is greater than it would be if the competitive inhibitor was not present. Therefore, while it reaches max, it levels off only with greater substrate concentration. Non-competitive inhibitors cannot be overcome with substrate, so an enzyme's effectiveness is lessened. So only those that have been inhibited with, have not been inhibited will function. So it can only be overcome if you add additional substrate, or sorry, additional enzyme. So that's what these graphs are showing you here. And again, the concept of reaching saturation point, an example of, let's say I can eat a total of 4 marshmallows in one mouthful, but I can't eat more than 4 marshmallows. So if I can eat 1 marshmallow and I'm swallowing that, I can eat 2 marshmallows at a time, I can eat 3 marshmallows at a time, I can eat 4 marshmallows at a time, but I can't eat 5 marshmallows at a time. If I put 5 marshmallows in my mouth, I'm only going to be able to swallow 4. If I put 6, I can only swallow 4, 7, 8, 9, to the point where it's just falling out. That's the point of increasing substrate doesn't increase the rate at which it works. It's like that. The active site's like a mouth that can only eat a certain amount, so it's not going to go any faster than that. Environmental regulation. So different temperatures and pH and what are changed. So again, this is why your body needs to maintain a pretty narrow range of both temperature and pH. So if it gets too cold, your enzyme's going to look normal, but the molecules are moving so slow that they're not going to interact very well. So at point Q, this is the ideal temperature. This is where the molecules are moving fast, the enzyme's still in its correct shape. At point T, the enzyme starts to denature, and at this final point here, the enzyme's fully denatured to the point where the enzyme doesn't work. For pH, only at point R is it actually functional. Everything else, you see, it loses its function due to denaturation. Notice I added this extra arrow just to show that it's denatured here or here. In temperature, only high temperature denatures an enzyme. Cold just makes it go slower. All right, so that concludes what I wanted to talk to you about with enzymes. So I apologize for the interruptions in between, but hopefully we can still have the basic understanding of what's going on.